Good evening, Samesh. I'm Rahul Saha, and today we'll be discussing about a very unique cycle known as the Krebs cycle. Good evening, Pratham. So today we'll be talking about the Krebs cycle. The Krebs cycle was discovered by Sir Hans Krebs. Okay, he was awarded the prestigious Nobel Prize for this specific discovery. Okay, this is also known as carboxylic acid cycle, like tricarboxylic acid cycle. Tricarboxylic acid cycle, that is the TCA cycle. Okay, and citric acid cycle. Sometimes it is also known as citric acid cycle. All the three names are there. I'll tell you why it's called by these names specifically. But let's get down to why do we need Krebs cycle in the first place. Again, coming on to what is the need of this biochemical cycle, what happens in this biochemical cycle, and what if this cycle has some mutations, this cycle does not go as per the requirements of the body, what could be the specific consequences of the happenings or the consequences pertaining to the dysregulation or dysfunction of this cycle. So coming down to Krebs cycle, why do we need it? The thing is, till glycolysis we have been able to produce pyruvate. Okay, so biochemistry is all about deep understanding. We have been able to synthesize pyruvate molecule, two pyruvate molecules from one glucose, 3 carbon each. So from enol form to keto form it was able to tautomerize. Okay, we have been able to make pyruvate, but we know that pyruvate still has a lot of energy. All the energy has not been converted. Okay, only 2 ATPs have been produced, but pyruvate still has a lot of energy remaining because the del G was negative 61 point something, right? And actual del G was minus 31.9 kilojoule per mole. So it has a lot of energy remaining in it, the pyruvate molecule still, and that can generate more and more energy. So one glucose molecule has only given us till now a net of 2 ATP molecule per one glucose molecule, but we can generate more energy from that. But in order to do so, pyruvate must get inside the mitochondria. The mitochondria is a double layered organelle. Okay, you have cristae like this, inner membrane, outer membrane. So outer membrane of mitochondria, the outer membrane when it comes to mitochondria is almost permeable to everything, very permeable. But the inner mitochondrial membrane is extremely selective when it comes to permeability, very selective. Okay. So what you have is a specific transporter for you can say pyruvate, that is known as mitochondrial pyruvate carrier, or specific MPC transporter. Okay, mitochondrial pyruvate carrier known as MPC. So this MPC transporter can transport pyruvate inside or into the mitochondria or this pyruvic acid. Once this pyruvic acid has been transported into the mitochondria, into the mitochondrial matrix, okay, it then undergoes a process known as oxidation followed by decarboxylation. Means one CO2 group from the pyruvate is taken out. So the entire thing is cumulatively known as oxidative decarboxylation, and then it is transferred or then it is converted into, it reacts with a specific molecule known as coenzyme A, and then it is converted to acetyl coenzyme A. Okay, I'll explain to you that reaction. And when acetyl coenzyme A has been generated, acetyl CoA is a very specific molecule. It is generated from the degradation of fats also. So that acetyl coenzyme A can enter the Krebs cycle. Okay, and the main event or the main aim behind our body going for Krebs cycle is not generation of ATP or GTP. Why the Krebs cycle keeps on going is it has a very serious reason. the main reason which is not given in many books, okay, regarding the Krebs cycle, but that is the most important reason, is generation of reducing equivalents. Generation of reducing equivalents, okay, which will reduce something and will oxidize itself, means which will transfer electrons, which are the carriers of electrons, like NADH, okay, like your NADH and FADH2, okay. So these are the, you can say, electron carriers, the reducing equivalents. If these are not generated in copious quantity, at enough or at appropriate amounts or concentration, I should be saying, then what will happen is that if you remember, I had told you that in the mitochondria you have complex 1, then 2, then 3, then 4, then complex 5, that is F1, F0, ATPase. So who throws electron from where? This NADH comes and binds to complex 1 and starts throwing electron from NADH to complex 1, then that electron goes from complex 1 to complex 2, complex 2 to complex 3 to complex 4, and while the electrons travel, due to the potential energy, the per electron travel 4H plus is pumped out, sorry, I'm drawing it just the opposite, 4H plus are pumped out from mitochondrial matrix to the intermembrane space every time. So what you generate is an electrochemical gradient. You have too many H plus here, too few H plus here. So this side will be acidic, that is the chemical gradient, and this side will be very, very positive compared to this one, because each H plus is carrying one positive charge. So electrochemical gradient, that is the electric gradient and chemical gradient. And when the H plus will eventually come back or return to the matrix via its electrochemical gradient to reduce the potential energy, so that potential energy will be harnessed to phosphorylate the ADP with Pi and generation of ATP. So if and only if these electrons do not flow, this electrochemical gradient can never be generated. And if electrochemical gradient is not generated, you will never be able to synthesize ATP, because once this H plus goes through the F1 F0 carrier via its potential energy gradient, okay, then only ATP can be produced. So the main aim of the experiment is what? To generate this NADH, to generate another one FADH2, which I will tell you what it is. It is also a brother of NADH only, flavine adenine dinucleotide, okay. NADH is nicotinamide adenine dinucleotide hydrogen. It is flavine adenine dinucleotide hydrogen 2. That is all it is. This is also a reducing equivalent. It can carry electron because it is H, right? The H is carrying electron. It is at the reduced state. So it will reduce others and oxidize itself. Okay. So these are the reducing agents that you have. That are the electron carriers, and they donate electrons very, very easily. They oxidize themselves and reduce others. So they are the reducing agents. So you need to generate this reducing equivalence. Otherwise, you can never go through this electron transport chain. And that is why the prime biochemical reason, understand it carefully, why you need, or why do you need Krebs cycle in the first place in the body, is to generate, good evening Arya, is to generate the reducing equivalent, is to generate the NADH and FADH2. That is why we need the Krebs cycle, okay. So now proceeding with the Krebs cycle, what we have is, we have an enzyme. That enzyme will cause this oxidation of pyruvate. Once pyruvate gets into the mitochondrial matrix, it will be converted from pyruvate to acetyl-CoA, okay. So the pyruvate dehydrogenase enzyme does this job, okay. So multiple copy of these enzymes are located in the mitochondria of all eukaryotic cells, and in the bacteria they do not have mitochondria, so it is located in the cytosol of bacteria. But in bacteria also, prokaryotes also, the Krebs cycle is well underway, right? Now this pyruvate dehydrogenase complex, okay, is a very, very special molecule. It's a very, very special enzyme, I should be saying. Why? Because what it does is, this is the reaction I'll tell you. Pyruvate inside the matrix or in the matrix of the mitochondria, okay, there is an enzyme known as pyruvate dehydrogenase complex. Dehydrogenase complex. Okay. So this enzyme is made up of three sub-enzymes. Okay. E1 plus E2 plus E3. The details of this is not there in your course, so I'll just leave it here only, E1 plus E2 plus E3. But know that this enzyme is not a monomeric enzyme. It is made up of multiple polypeptide chains. Okay, that's why E1 plus E2 plus E3, and those polypeptide chains combined, when they combine cumulatively, then they make this specific enzyme. So this is a very, very complex enzyme. Okay, that is the perspective here. So what happens is you have pyruvate. So pyruvate's structure is this one. If you know the structure that I had drawn. So this is the structure of pyruvate, and what you add is you add coenzyme A carrying the thiol group or sulfur group. Okay, since this is an oxidation reaction, something must be reduced, so NAD plus is reduced, 2 NADH, 1 NADH you generate here. Okay, and then you have the different cofactors. You have thymine pyrophosphate, you have lipoate, you have TPP is thymine pyrophosphate, vitamin B1, then you have lipoate, and you have FAD also. Okay, all these things, what they'll do is they will liberate one carbon dioxide. Okay, and what you will get is you will get the C, and the O will stay, this O will stay. Okay, so CO, then this acetyl CoA will come here. Okay, this is CH3. Okay, and this CO2, this group, okay, this group got liberated. So CO2 got liberated. So this is an acetyl group. This is the coenzyme A. So now this molecule is known as acetyl CoA. Very, very important molecule, acetyl CoA. Okay, the del G of this reaction is minus 33.4. The del G not at equilibrium. Okay, minus 33.4 kilojoule per mole. So it's a very, very spontaneous process. It's a oxidation process, oxidative decarboxylation, because carbon dioxide is released from the molecule or the substrate known as pyruvate. So note down this reaction very, very carefully. This is the complete reaction, how one pyruvate molecule becomes one acetyl CoA. But you have two pyruvate molecules generated from one glucose, so just multiply it by 2. So you will need 2 pyruvate molecule, 2 acetyl CoA, and everything will be doubled here. Okay, the requirements: 2 NAD plus, 2 NADH, 2 CoA, SH, okay, 2 TPP, 2 lipoate, and 2 FAD. So they will be all required to be doubled. But the thing is, this is oxidative decarboxylation, and in the end you produce this specific molecule known as acetyl CoA. Okay, so the pyruvate dehydrogenase complex in turn uses 5 coenzymes. So I had told you that an enzyme is inactive and it is not bound to coenzymes and cofactors. It is known as apoenzyme in the biomolecule chapter. When it binds to the coenzymes and cofactors, it changes its conformation. Its active site becomes very, very stable and can, you can say, bind the substrate very, very tightly, thus lowering the activation energy, and then becomes very, very active, completely active, known as holoenzyme. Okay, so the combined dehydrogenation and decarboxylation, means the redox reaction and decarboxylation of pyruvate to the acetyl group of acetyl CoA, that requires a sequential action of 3 different enzymes, that is E1 plus E2 plus E3, and 5 different coenzymes, also known by the name in biomolecule chapter prosthetic groups, because they are not the real, you can say, groups which belong to the enzyme. Okay, number 1 is thymine pyrophosphate. The 5 coenzymes can be asked in the NEET examination. Okay. Number 1 is thymine pyrophosphate, this TPP. I'll write the full form, so I'll erase it. So the coenzymes required by the enzyme PDH, we'll be talking about pyruvate dehydrogenase, are: number 1 is thymine pyrophosphate. Thymine is vitamin B1. That is TPP. Okay. Number 2 is flavine adenine dinucleotide. Okay, FAD. Adenine dinucleotide. Okay. That is FAD. Then we have something known as coenzyme A, okay, which I denoted with COASH because it has the thiol group or sulfur group. Okay. Third one was coenzyme A. Then I had written also nicotinamide adenine dinucleotide, NAD. Nicotinamide adenine dinucleotide in that reaction I had written, right? That is the NAD. And in the end, don't forget the lipoate. Okay. Lipoate. So the thing is, why does the doctor recommend us to take vitamins? Because of this only. Because if thymine is not there, then thymine pyrophosphate will not be produced, vitamin B1, and if we have vitamin B1 deficiency, then this acetyl-CoA will not be produced, and then what will happen? It will not get into the Krebs cycle, and NADH more, and here only one NADH has been produced and more NADH are produced in the Krebs cycle only, and if NADH are not produced, you will not get ATP. So in simple, in a layman terms way, you might have heard that vitamins give you energy. So vitamins do not give you energy. They activate enzymes of the Krebs cycle so that your ETS cycle can be regulated properly, which starts after the Krebs cycle, after the generation of the reducing equivalents which has been generated via the Krebs cycle, and then ATP can be produced via the F1 F0 or complex 5 ATPase, right, present on the inner mitochondrial membrane. So 4 vitamins are required for this specific continuation of this vital component known as the Krebs cycle. Number 1 is thymine. Okay, thymine for, you can say, thymine pyrophosphate. For this one you need thymine. Okay, vitamin B1. Okay, water soluble vitamin. Then what you need is, you need also riboflavin, vitamin B2 for fat. Flavin means flavin came from riboflavin. Okay, so this FAD, okay, came from riboflavin. Okay, B2. Then we have niacin. From niacinamide came NAD. Okay, vitamin B3. And then we have pantothenic acid, vitamin B5. From that comes coenzyme A. Okay, so coenzyme A comes from pantothenic acid, vitamin B5. Pantothenic acid, vitamin B5. So these are the vitamins which are heavily needed by your body if you want to continue the Krebs cycle and you want to stay alive. Because if the Krebs cycle is completely ceased, what will happen is, without ATP the cell will die. Only glycolysis has 2 ATP that you get from glycolysis will not be able to generate more and more, you can say, sustenance for your body for a longer period of time. You will eventually succumb to death, right? That will happen. Yeah, Spidey. So Spidey one, Samesh, the thing is, when you will learn genetic engineering, you will learn that we cannot just transform the entire genome. What we can do by the current technology right now is we can insert some specific genetic sequences. Okay, that's the maximum we can do, and we have to check in the human body what are the effects just by inserting, and don't think that that protein will be made and we will start jumping. Each and everything must undergo a certain thing known as clinical trial that you might have heard of. So clinical trial, once you go through a clinical trial or once you put a human being through a clinical trial, there are a lot of ethical issues. Okay, you might have heard last year only a Chinese person he knocked off the CXCR4, which is the chemokine receptor to which the HIV-1 virus binds. Okay, that GP120 binds, glycoprotein of 120 kilodalton, the ligand. But the thing is, if CXCR4 is not present, the T cells, the other receptors, okay, the other cells may not be able to interact. The immune system may be weakened. Okay, so the thing is that scientist was put in jail. Okay, so again, it is providing immunity to HIV for life, but it is providing a myriad of side effects also along with it. Okay, so we need to be very, very careful when we go for any kind of genetic engineering experiment, and especially these kinds of, you can say, very, very fascinating and super ambitious projects where we think of putting a specific protein in the body for which there is no receptor. The protein only will not make you Spider-Man, right? The protein will need its receptor, the entire pathway, signaling pathway, because after the generation of the protein, what will you do? You should be able to communicate with the cells also. Okay, so you will need to put the genes for the receptor also, and then if those receptors use up or interfere with any other pathway, any other signaling cascade, then what are the specific ramifications, clinical ramifications? Because that is spider and we are humans. You are inculcating something in us, it may disturb any other pathway also. Then what are the ramifications? So those things we have to check very, very properly in order to even think of carrying out such kind of experiment, right? So after you have these 4 types of vitamins, okay, what we have is, you can say, we will now talk about this coenzyme A. So coenzyme A, I told you, is a reactive group which has a thiol group, SH group. It is very critical for the role of coenzyme A. It is an acyl carrier you can think of in many metabolic reactions. Okay, it forms thioesters. Thio means sulfur again. Okay, and it has a very high standard free energy. Okay, so they have very high acyl group transfer potential you can think of, which can donate the acyl group to a variety of acceptor molecule in a very, very favorable reaction type. Okay, the 5th cofactor of the PDH complex, the lipoate. Okay, the lipoate that I written has 2 thiol groups that can undergo reversible oxidation to a disulfide bond. This one, SS, similar to cysteine disulfide bond. This one, SS, the lipoate can undergo 2 reversible oxidation reactions. Okay, now I'll just tell you the name of E1, E2, E3. No need to know the entire structure of it. So the name is number 1 E1 because the pyruvate dehydrogenase complex. Okay, pyruvate dehydrogenase complex, okay, PDH, is made up of 3 enzymes. Okay, this enzyme is made up of 3 sub-enzymes, you can say E1, E2, E3. So E1 is nothing but pyruvate dehydrogenase only. So E1 is known by the name pyruvate dehydrogenase. Okay. Now coming to E2. So E2 is known by the name, you can say, dihydrolipoyl transacetylase. Just the name I am telling you. If you want to remember, you can, but just for your knowledge purpose, this I am telling you. This will not be asked in the examination. Okay, transacetylase. These 3 names I thought I should tell you in order to keep the concept clear. Then E3 is dihydrolipoyl dehydrogenase. Okay, transacetylation, dehydrogenation reaction will happen. So these are the 3 specific sub-enzymes present in multiple copies, multiple polypeptide chains, you can say, to make a huge complex known as PDH. Okay, so if you just think of, okay, you have 60 copies of only E2 in a bovine enzyme of PDH. You can say in the bovine model you have 60 polypeptide chains of E2 only in one PDH complex. So think of how big the PDH complex is. Okay, it's a huge complex, extremely huge complex. Okay, so that is your pyruvate dehydrogenase complex. And now then we can start now the Krebs cycle. So once we start the Krebs cycle, okay, so once we start the Krebs cycle, what we have in the beginning is, you can say, the acetyl-CoA group. Now the acetyl-CoA has already entered the Krebs cycle. Now once acetyl-CoA has entered the Krebs cycle, it will react with oxaloacetate. From where oxaloacetate come, I will tell you. This entire reaction is given in your book. So this acetyl-CoA reacts with oxaloacetate, okay, and forms citrate. So I'll just show you the reaction. Okay, first step is acetyl-CoA you have been able to produce now. Now oxaloacetate OAA will come plus this one. Okay, you have an enzyme known as citrate synthase. Synthase means does not require ATP. Synthetase would require ATP. This is synthase. And what you get is, you get citrate. That is why it is also known as citric acid cycle, Krebs cycle, because of Hans Krebs, citric acid, because the first intermediate is a citrate. And why tricarboxylic acid cycle is, this citrate contains 3 carboxylic acid groups. I'll just draw the structure and show you. Okay, you have CCH2, CH3, or CH2, CH2, here CH2. So one hand of CH2 is free. Now what I can add is, I can add OH here, one hand, this one free, CO minus, CO minus, CO minus. So now I think the valency is completely done. Now just think of how many carboxylic acid groups, CO, 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 you can add HHH. So 3 carboxylic acid groups are there or not? Okay, 3 carboxylic acid groups. That is why it is known as a TCA cycle, tricarboxylic acid cycle, because the first intermediate is citric acid or citrate. A toric acid is same in biochemistry. So citric acid cycle. TCA means 3 carboxylic acid groups are present in citrate in the first intermediate only. It is known as the tricarboxylic acid cycle. And since Sir Hans Krebs was the discoverer of this reaction, this specific cycle, it is known as Krebs cycle, in order to pay tribute to his name. Now drawing the structure of acetyl-CoA, okay, we all know that CH3CO, then SCOA. This is acetyl-CoA. Oxaloacetate, what is the, you can say, formula? It is a 4-carbon compound, okay, made up of CO. Okay, 1, 2, 3, 4 hands here, then CH2CO. Okay, this is oxaloacetate, 4-carbon compound. Citrate is how many carbon? 6 carbon, because here 4 carbon, here 2 carbon. Check here, carbon stoichiometry again. 4 carbon, 2 carbon. Okay, always check these things. Carbon number 4 plus 2, condensation reaction. Okay, combination is 1, 2, 3, 4, 5, 6. Now you have 6 carbons here. Okay, so that is again the biochemical stoichiometry balancing. Just like you balance a simple chemical equation, we balance biochemical equation also. Okay, just count the carbon number. That should not be mismatched. So 4 plus 2 carbon, you get 6 carbon here. Okay. So after this reaction is done, what you have is you eliminate the coenzyme A molecule. Okay, you add water and you eliminate coenzyme A. Okay, so the COASH gets eliminated. Because you can see there is no COASH in this citrate. Okay, so water has been added, COSH has been liberated. This COASH is liberated, this H is taken in, and this OH is put here. Okay, now SHOH. So this H2O has been completely used up. Okay, see here, it will be one will go to a 1H, and this OH here. This complete H2O has been used up. Okay, so this is the first reaction of the Krebs cycle. Okay, the first reaction of the Krebs cycle is this one, formation of citrate molecule. From the reaction, condensation means you have something which gets liberated, that is coenzyme A. Condensation reaction between the acetyl CoA and the oxaloacetate. Okay, now there will be a dehydration reaction, okay, where OH group of citrate is repositioned to isocitrate. That is known as dehydration, then rehydration. Means first OH group is taken out, then the same OH group is put in some other place. Okay, in the same molecule, that causes it to go for decarboxylation in the next step. Okay, so this is known as, you can say, cis aconitate. So the next reaction will be, you can look at your book also, you have citrate here. Okay, so this will be a reversible reaction, del G will be almost positive. Okay, there will be a molecule known as aconitase, it will break down the OH and put OH again back. Okay, and what you will form is cis aconitate. What you form is cis aconitate. Now what is cis aconitate? Cis aconitate is nothing but, you can say, CH3 CO minus. This side upward you can put CH2 CO minus. Okay. Here what you can do is you can put a double bond, CHCO minus. Okay. So now what I have done is I have taken out water from here or not. I have gone for the dehydration step, because here there was OH in citrate. If you see the citrate structure was like this: CH2 CO minus, okay, COH CO minus, and then you had CH2 CO minus. Okay, so now I have taken this H2O from here, okay, and then 2H has been taken, 1 is remaining and 1 O. So H2 has been taken out, okay, from here. Here it is dehydration. Okay, and this intermediate has been produced, which is known as cis aconitate. Okay, this is cis aconitate, this one. Aconitate. And the cis aconitate gets converted into, you can say, isocitrate, again rehydration, okay, but with changed position. Now what will happen is, rehydration happened, okay, rehydration has happened, but position has been changed. How? You have CO minus, just look at citrate and isocitrate. You have CH2 CO minus. Instead of putting OH here, I'll put H here only. Okay, and what I'll do is, in isocitrate, in the third carbon, I'll put OH. In the third carbon, I'll put OH here. Okay, and when I put the OH in the third carbon, this side will be H and this side will be, you can say, CO negative. So that can be done. CO negative. So this is what your isocitrates, or this is how your isocitrate looks like. Okay, so you can just check the entire thing has been changed. It was from the first carbon to the fourth carbon. It has been, you can see here, it was first carbon OH, here it is at the, sorry, carbon number 1, so carbon number 2, here it is in the carbon number 3, you can say, carbon number 3. Okay, so carbon number 3 has got OH, earlier it was having OH in carbon number 2, so the position of the OH has been changed. And why you are saying that, you will think that, sir, why do you need to change the position of the OH? You are going for this complex reaction, or the body. I am not going for this complex reaction. This complex reaction happens in the body only. Okay, so you change citrate into isocitrate. The intermediate is this one, cis aconitate. The body goes for this complex reaction. Why? Because you need to go for decarboxylation reaction after this. So it sets up the decarboxylation reaction properly. That is why the body goes for this decarboxylation reaction. Okay, so radioactive spider biting will not have any problem, Pratham. That is not a problem at all. Okay, so if the radioactivity is transferred, if it bites for so long that the radioactivity gets transferred, then only you may be having some or the other kind of problem. Otherwise, you wouldn't be having any problem whatsoever. So the thing is, the next part of the reaction is, now it is known as oxidative decarboxylation reaction. Now what will happen is, this isocitrate would be converted into, or would be catalyzed to become alpha ketoglutarate. Okay, now this third reaction is isocitrate becomes alpha ketoglutarate. Now what is this reaction all about? Now just imagine that, draw the structure of isocitrate. Okay, take out decarboxylation, or you can say CO2, that is oxidative decarboxylation, because dehydrogenase is there. So dehydrogenase will go for, you can say, redox reaction. That is the work of decarboxylation. Okay, they will go for redox reaction, oxidative decarboxylation. Now once we have oxidative decarboxylation reaction, so in the end we have to go for NADH, because NAD plus will be reduced for something to be oxidized. So NAD plus will make NADH. That is the first step of why Krebs cycle is being done, in order to produce NADH and fat H2. So here NADH is being produced. Okay, so we draw the structure of isocitrate again. Isocitrate is C, CO minus, then CH2, CO minus, and you will be having H here, then OH here, okay, and then H that side, and CO minus. This is the isocitrate. Now you take out CO2, you have this dehydrogenase enzyme. When you have dehydrogenase, means one NAD plus will reduce itself to NADH and H plus. This is known as reduction of NADH, because this is an oxidative decarboxylation reaction. This is an oxidative decarboxylation reaction, same as the reaction which I showed you, which was happening in pyruvate. So pyruvate was catalyzed to acetyl-CoA. That was the oxidative decarboxylation. Okay, here it is again, oxidative decarboxylation. Okay, so now oxidative decarboxylation means what? Oxidation, something has to be reduced, so NAD plus is reduced to NADH plus H plus. Now you have got alpha ketoglutarate. Now this is the first intermediate reducing equivalent that it is needed, that is needed by the Krebs cycle. So alpha ketoglutarate looks like this. Okay, it will have a center CO. Okay, on top I can put CH2, then CH2CO minus. Okay, then downward COO minus. Okay, so you can see that 1 2 3 4 5 were here. Okay, 1 2 3 4 5 6. Now 1 2 3 4 1 2 3 4 5 carbon is remaining. So one carbon has got out. Okay, one carbon dioxide has got out. Okay, you can see clearly here one carbon dioxide has got out. So this CO2 has been eliminated completely. Okay, the CO2 has been eliminated completely. That is the specificity of that specific reaction. One CO2 has been eliminated, and that is why we say that people say, you also say that we exhale CO2. The question is, from where did CO2 come? Where did CO2 come from? Where did CO2 originate? Okay, or where was its origination? The origination of CO2 is in the Krebs cycle. You can see first CO2 coming out from here. You had seen that oxidative decarboxylation, first one more CO2 had come out where when pyruvate was, you can say, converted to acetyl coenzyme A. So now you can understand respiratory tract once you have read the Krebs cycle. That is why biochemistry is the root of biology. So in respiration we are taught since class 6 that we exhale CO2, but from where did CO2 come? When pyruvate gets inside the mitochondria, first of all pyruvate is oxidized to acetyl-CoA, oxidative decarboxylation there again, so decarboxylation happens there, CO2 coming out there, and here also CO2 is getting released. So emanation of CO2 happens inside the mitochondria via with the inception of the pyruvate oxidation and then acetyl-CoA formation, and acetyl-CoA joining the Krebs cycle or getting into it is being subjected to tricarboxylic acid cycle. So that is how we generate this specific alpha ketoglutarate. Okay, second thing, okay, we go for one more time. Okay, what we do is one more time we go for something known as oxidative decarboxylation. Now here what happens is this alpha ketoglutarate will be converted to succinyl coenzyme A. Now again coenzyme A will come, okay, and will replace one CO minus group. The CO minus group will be taken, the CO2 group will be taken out again, one more carbon dioxide taken out, once more oxidative decarboxylation. This is an oxidation process. CO2 is coming out, it is being liberated. So again you have decarboxylation. So oxidative decarboxylation means that you will need one more time. Okay, you will need one more time the release, or you can say the usage of NAD plus, because if NAD plus is absent, what will happen? There is no acceptor of electron. When there is no acceptor of electron, the entire system may become unstable. The electron can go and bind to some other molecule and may, you can say, reduce it and cause, you can say, n number of damage in the cell. It can bind to the DNA, it can bind to the lipid. Okay, so you need some specific molecule to quench that electron. That is why it is redox reaction, oxidation and reduction side by side. So what happens is this alpha ketoglutarate, the next reaction is this alpha ketoglutarate. One more time enzyme will be dehydrogenase. Okay. Now CoA SH will come, CO2 will be released. So this is again oxidative decarboxylation, and since CoA group has come, it will become succinyl coenzyme A. Okay, let's draw the structure. Okay, here NAD plus will become, it will be reduced to NADH plus H plus, since this is an oxidation process. Okay. So alpha ketoglutarate structure I had drawn already. I'll draw it again. CO. Then on it will have 5 carbons, CH2, CH2COO, COO, CH2CO minus. Okay. Then downside will be again 1, 1, 2, 3, 1, 2, 3, 4, 5. Okay. COO minus. This is alpha ketoglutarate. Now what will happen is one CO2 will be removed once more, and you will be having coenzyme A group added. Okay. So what will happen is now to that place where double bond oxygen was attached, coenzyme A will be attached. CSCoA. Here double bond oxygen, here it has been replaced by coenzyme A. Okay. Then downward you will be having double bond oxygen. Fine. Then upward CH2CO minus. How many carbons? 1, 2, 3, 4. Now the fifth carbon, this one is gone. You can say C, 1 O here, 1 O here. So O2 is eliminated. This one. Okay. So now you have only 4 carbon here. You can see this one was 5 carbon. Whereas the isocitrate, the citrate was of 6 carbon. So now you have a 5 carbon compound. You have one more CO2. One more NADH, so you have generated 2 NADH in Krebs cycle till now. If you do not count the NADH which was generated from pyruvate to acetyl-CoA conversion, in the conversion where pyruvate was converted to acetyl-CoA, we have to count that also. But in the real Krebs cycle, if you check from acetyl-CoA only, 2 NADH has been generated. One from isocitrate, 2 alpha ketoglutarate oxidative decarboxylation, 1 dehydrogenase enzyme, again a dehydrogenase enzyme causing oxidative decarboxylation of alpha ketoglutarate to succinyl-CoA, and here you have added one coenzyme A group also, so it has become succinyl-CoA. So this molecule is succinyl-CoA, and here you have got 4 carbons, here 5 carbons. You are losing 1 and 1 carbon each. Okay, so what you are doing is, in pyruvate molecule, how many carbons did we have? We have 3 carbons. All those 3 carbons will be released. So this is another way of looking at the Krebs cycle. So all the 3 carbons present in the pyruvate or pyruvic acid skeleton is being now released and broken down. So we are utilizing the entire energy that I was talking about present in the pyruvate molecule. So till now 2 carbons have already been removed in the form of carbon dioxide. Okay. So we are inhaling that thing and we have used the entire energy present in the pyruvate molecule. Okay. So first CO2 was taken out when pyruvate was changed to acetyl-CoA. Second was taken out when isocitrate has been oxidatively decarboxylated to alpha ketoglutarate, and the third CO2 has been taken out or has been liberated when alpha ketoglutarate is again oxidatively decarboxylated to succinyl coenzyme A. Now the entire pyruvate skeleton has been broken down and all the energy has been extracted from pyruvate. So that is the one of the main reasons again behind the continuation of the Krebs cycle. Okay. So this is how the Krebs cycle works in the first place. So till now we'll stop it because this is the entire concept. The rest half of the Krebs cycle we will be talking about in the next class. So I hope you are able to comprehend the lecture. If you have had any doubt, you can ask me directly on my personal number and I'll clarify that doubt for you. Okay, then thanks a lot and we can continue the lecture in the next class. Thank you.